Hi, everybody. I wanted to just make a quick video for you about some things that I learned about the virus and some medicinal chemistry efforts to develop treatments for it. So as you might know, the new coronavirus is a so-called single-stranded RNA virus. That just means that its genome is not DNA, it's RNA, and it's single-stranded RNA. There's a lot of different viruses that have their genome sort of organized in different ways. So I've sort of represented what the genome looks like here as a single-stranded nucleic acid with the various RNA bases. And then what the viral RNA genome really does is two things. Number one, it needs to replicate itself. And then it also, the single-stranded RNA just serves as a messenger RNA to make viral proteins that then make the viral sort of capsule, and then the assembly of which makes new virus. So what's kind of interesting to me is there's been a lot of there's been a drug that is used that is showing some effects for the virus, and the drug interferes with this process, which is the replication of the viral genome. So I'm going to actually scoot you down and I'm going to show you something about how that works, if you don't mind. Let's see how this goes. Okay, that's pretty good, I think. Okay. So down here, how the viral genome gets copied is pretty straightforward. The single-stranded RNA directs the polymerization of nucleotide triphosphates according to so-called, Watson-Crick-Franklin base pairing. So if this is the genomic DNA, it will sort of template the polymerization of a complementary strand, which then, of course, can make more of the viral DNA strand, or I'm sorry, not DNA, RNA strand. So let me tell you now about how this drug works, if you don't mind. I'm just going to flip this over real quick and raise this back up. Here we go. Sorry about this, production quality. Okay, so I wanted to zoom in on one of the nucleotide triphosphates that's one of the building blocks for the polymerization reactions to make the RNA that's the viral genome. So I picked ATP as an example. So the adenosine part with one of the phosphates is what's incorporated into the growing strand. And right here below it is the structure of the drug. It's called, I don't even know how to say it, Remdesivir. And I want you to notice a couple things about Remdesivir. One is that it is sort of a synthetic version, an altered version of a nucleoside. So it resembles adenosine. You can see that there's a ribose ring. It has that sort of like the same purine ring. It has this weird group attached at a position where there is no group in ATP. Then, interestingly enough, the phosphate group has this other organic sort of groups attached to it. That's because ATP itself is an ionic compound. Ionic compounds are just generally very poorly absorbed as drugs, and they don't cross the cell membrane, so they're just not very useful. So these groups that I've circled in red on the remdesivir structure, those basically keep the drug neutral when the drug, so that it can enter cells. When it enters cells, these groups are cleaved off, so those are removed. Any drug that works that way That's called a so-called prodrug, and then upon some metabolic activation, it gets turned into the actual drug. So remdesivir actually has to have two things happen to it. It has to have those groups removed, and then it has to have two phosphates added, because the sort of starting material for RNA synthesis are nucleotide triphosphates. So both of those things happen. These groups get removed, and then the cellular sort of enzymes add two phosphates. So now this remdesivir triphosphate looks very much like adenosine triphosphate, which is the natural starting material for RNA polymerase, which is the enzyme that makes the viral genome. So let's call this molecule, this synthetic molecule, RTP as opposed to ATP. So how the drug then works is it gets into a virally infected cell, and let's say that the RNA polymerase machine is laying down the complementary base, so like a U opposed to an A C opposed to a G, etc. Every once in a while, when what's called for is an adenosine group that's paired to a uracil, the enzyme will make a mistake and mistakenly incorporate remdesivir. When that happens, it causes all kinds of problems with that particular RNA molecule that's now sort of like a poisoned genome, which then sort of has cascading effects for viral replication and the viability of the virus. So that's sort of how this compound works again, that it's an analog of the natural substrate ATP. It's a prodrug that has these groups cleaved inside of the cell and activated as the triphosphate, and it's actually mistakenly incorporated into the biosynthetic. It's incorporated into the growing RNA strands of the virus, and then it makes that sort of poisoned RNA molecule non-functional. So what's interesting about remdesivir is doctors have been using it. There's one case in Washington where a patient was in the ICU, and the doctors were able to use remdesivir under so-called compassionate use guidelines, which means if a patient is not doing well, you're allowed to try sort of off-label drugs and to see if it would help. And anecdotally, the patient started improving the following day. So, it's very early, but this molecule is in clinical trials now, here in China and in some other places, to see if it improves the outcome for patients with the virus. The one bad thing about remdesivir that you can probably tell from its structure is that it's very complicated. It's very difficult to make this compound. It's probably very expensive to make this compound. So those are, even if it does work extremely well, I think its availability might be quite limited for some time. But anything that I think we have to help sort of blunt the effects of the virus against people that get affected very, very severely by it would certainly be really, really helpful. All right. It's always nice talking to you.